Okay, how's it going, sixth graders? So this is just going to be a podcast that's going to cover the three types of Punnet squares and just simple ways of how you can identify them and how to solve each one. I saw a lot of your ears bleeding when we were going over some of this stuff. It can get kind of complicated, so just watch this. If you're not understanding it still, just rewatch the podcast until you get it. Again, we'll have a review tomorrow, and I will have review sessions during lunch, so you can come with me with any questions you have. So the first kind of Punnet square we're going to do is we're going to do the recessive dominant one. Recessive dominant one. And the recessive dominant one, remember, it's got like the dominant allele and it's also got the recessive allele. Again, it can be any type of letter, but you always want to keep the letters the same, and just lowercase indicates recessive, uppercase indicates dominant. I saw a lot of you were trying to say stuff like, oh, well, what happens if the recessive allele is mutated and becomes, can mask the dominant one? Well, that wouldn't work in that case. That R, if it was mutated, would eventually become the dominant trait and the recessive trait. All we're saying with the dominant recessive is if you're uppercase and dominant, you will always cover up the other allele. That's recessive and lowercase. If it was a mutation and this one decided to start covering up this one, they would switch places. This would become uppercase and it'd be dominant. This would become lowercase and recessive. So don't confuse that. Remember, dominant alleles, recessive alleles. Now when we're doing these kind, the easy thing that'll tell you that you're just dealing with a simple recessive dominant Punnet square is if you see right there a recessive and a dominant trait. So we look right here and you see there's your dominant trait. Uh-oh, here comes your recessive traits popping in. So easily that tells you this is a recessive dominant Punnet square. So we come through here and we fill it in. So let's fill these in. Fill these ones in. And you get big R, little r, little r, little r, little r, little r, big R, little r. So then I'm going to ask you the question. I'm going to say, please tell me the phenotype. This line should be there, so let's just say phenotype. So I say, please tell me the phenotype. When we say pheno, remember we're saying physical. What do they physically look like? Let's say that right now we're measuring for, let's say big R is right-handedness and little r is left-handedness. Let's just use that for instance. That's not really how it's controlled, but we're just going to do a fun little example with that one. So we look here and we say, how many people can we notice that are right-handed? And we look right here and we go, oh, there's 2 out of 4 right-handed, so that'd be 50% use the right hand. What do we actually see? Well, that also tells us that 50% are left-handed. They're recessive, so 2 R's would mean left-handed. So we say 50% right-handed and 50% left-handed. Now, if we were to mix it differently and we were to have a big R, little r like this, it would change a little bit. You can see how the alleles would change. Then over here, you have a big R, little r, and you just have 2 little r's. So that's if this changed to a big R instead of a little r. Then all of a sudden your phenotypes would change a little bit. You'd have, as you can see, 3, 1, 2, 3 right-handed people, because remember that dominant one masks. So this right-handedness masks the left-handedness. So we have 3 different things out of the 4. So that would give us 75% are going to be right-handed, whereas you only have now 25% who are going to be left-handed. So 75% right-handed, 25% left-handed. Now, we can take this a step farther from phenotype. Yes, we can do phenotype, and phenotype is really nice to have, but sometimes you need to know exactly what their genes are. Are they a carrier? Do they have no possibility of passing it on? So what you do now is you come up with something called a genotype. The genotype is simply this. We look here, 25% of this type of genes, the big R, 2 of these, so that's 50% of the big R, little r, and only 1 of these of the RR, making 25% of the Punnet square. You pretty much just look at the genes inside of it and say, okay, well this one's different from all these, so it's its own 25%. These 2 are different from these 2, so their own 50%. And this one right here is different from the rest of these 3, so it's its own 25%. Now you wouldn't say something like, oh, 25% little r's. You would say something else. You'd say 25% homozygous recessive. This one you would call So we're measuring for this one right here. So I'll draw a little arrow so you can see. And again, this one too. Homozygous dominant. And this one would be called heterozygous. And I'll explain to you what they mean in a second. So, homo, as we know, means same. So we look here and we see homo means same. We also know that hetero means different. Now, for all our sakes and purposes, we're just going to say zygous would just simply stand for something like alleles. So if I say same alleles, well then you know that the alleles have to then look the same. They both have to be big R's. Both have to be the same. Same alleles have to be both small r's. So you remember homozygous and homozygous. Now, just because you say homozygous, as you can see, we have two different types of homozygous. You can either be dominant or recessive. So that's why you add on that little thing at the end. So you say homozygous dominant for this one, homozygous recessive for this one. Now these ones are different because these aren't the same. So you want to use that other word. Instead of homo, you say hetero, which means different. So you say heterozygous, so different alleles. And you don't have to refer to them being dominant or recessive because you know, because they're different, that they're two different ones. So that one right there would be your recessive dominant, and this is how you'd say genotype, and you already know how to say phenotype. You just, what are you looking at? What percentage do you look at? You don't really care if somebody's heterozygous versus homozygous with the dominant ones. You just only care about what they physically look like for phenotype, but you start to care about what alleles a person has if you're dealing with a genotype. Now, moving away from that, we run into the next kind of Punnett square called an incomplete. It's incomplete dominance. In an incomplete dominance, you'll get, the way you can easily identify those is all of a sudden you see alleles that look like this. Big R, times a big W. Because in incomplete dominance, there are no recessive, there are no dominant alleles. They're all equals. So incomplete dominance, they are all equals. And the easiest way to identify that is also to say, hey, wait a second, all these are exactly the same. And also it'll give you a clue, it'll say, oh, it's incomplete dominance. So if you ever see one that says, because I mean you could have the case where you can have somebody with all dominant alleles going with somebody with all dominant alleles, but they also, the other clue that you'd see is they wouldn't be different. Remember, when we're dealing with dominant and recessive traits, we're dealing with they're all going to be the letter B. They're a big B or a little b. But in these cases, all of a sudden you see these two same R's, but then what's this? You see these aren't R's anymore. These are W's. So in incomplete dominance, you'll then take these. So let's say RR is red. This R codes for the red color pigment in a flower, and this W codes for the white. So you have a red flower and a white flower. Well, we do our Punnett square, our incomplete dominance Punnett square. We have R here, W here, W here. And you're going to get all the same if you notice. So you get red, white, red, white, red, white. Now, in the other one, we'd normally be looking for, like if these are recessive and dominant, you'd be looking for, okay, which one's dominant, that one's going to show. But in incomplete dominance, because they're equals, they both show together, so they do a mix. They mix together. And so all of these you're going to see in the phenotype 100% pink. All these will mix the red and the white together and you'll get 100% pink flowers. Now, the whole thing is if I asked you what the genotype was, what genes are in there, you'd say, well, their genotype is 100% RW. All of them have this RW in there. You're not saying that they're pink. You don't say genotype's pink. You say genotype is RW. All their offspring is RW. Just like their genotype up here was RRWW. Now, what you can do is you can then take this RW plant that you just created, and you can mix it with a red plant. Because say you're getting sick of pink flowers. Well, you mix that RW with the RR, you mix the pink rose with the red rose, and all of a sudden you'll start to see this. 50% of their children will now go back to being red, whereas 50% will stay pink, so you can get more diversity. If you want white, well then you just mix it with W. and you'll get 50% to turn back there. And then you just keep breeding these ones if you just want to turn them all back to red and just kill these ones off. So that's something that you can do, and you can actually do this with flowers. And this is how you can see that flowers can change colors, but then populations can change back. And so that's how you're dealing with, like I said, incomplete dominance. Now, this next one is going to be called codominance. And if you remember, I was saying that if you work with your coworker, like I have some teachers that I work with, I can't fire them, I can't tell them what to do, and also I don't mix my science into their class. I never walk into Miss Pendleton's or Miss Delp's or Miss Jenkins and start teaching science during their class. No, that's their classroom, because we're co-workers. So it's important to remember that term co-workers, because that co in there just means it's equal, and we have our own space that we do our own things. The same thing can happen with different colors in animals. So let's say we're going to take birds, and these birds exhibit codominance. Now these birds, let's say we want to know what color feathers they are. So one of these birds we have is a black bird, and another bird we have is a white bird. All of the black bird's feathers are black. That's what gives it its black color. So we're going to use these F's to indicate their feathers. And again, the white bird, he also has feathers, and these white indicate its white feathers. Well, if you mix these together, that'd be very confusing. We need to show, that this is a black feather and this is a white feather. Well, instead of writing out black feather, that'd take a lot of time, we can instead just do this. Take the B, and drop it down here, and just put the B there, and put the B there. You can put the W here, you can put the W here. So this indicates the feathers are black. The feathers are white. Let's cross these two together now. So we have our white bird, we have our black bird, feathers are black, feathers are white. So we make our Punnet square, we fill it in, so let's put the black bird on top. Feathers are black. I hope you're doing this as we go along. You should be doing this with me. These feathers are white. So let's drop them down. So remember, these drop down. And we put these ones in, the white feathers. Now what we see here is we have only one type of bird now. We have 100%. You're going to get birds that have feathers that are black and feathers that are white. These are not going to mix. Mix? No. No mixing. Instead what you'll see is you'll see a bird. Here's my terrible bird again, for any of you who want to suffer through it. Wow, that's a really bad bird. It's an ugly bird. So here we have our ugly bird. And instead of being a gray color, or the black blocking out the white that we see in other ones, instead we'll see spots of black and we'll see spots of white, spots of completely black and spots of completely white. And that, my friends, is called codominance. So let's take a little quiz. Let's see how much you guys have learned. Let's see what we have here. Okay, so right here I have a 1, a 2, and a 3. These are 3 different types of Punnet squares. Now you guys are all experts at doing Punnet squares. You'd have no problem filling it in, but I want to see if you guys can identify, just by looking at these parents, so these are all P generation parents, just by telling me what type of Punnet square are we doing in each of these. So we know, do we mix the colors together? Do we have one that's dominant over another one? Or do we share spaces through spotting? So the first one I'm going to ask you is which one of these 3 will actually show spotting? Which one of these will not overpower the other one or mix with the other one? If you said 3, then you're correct. And again, the way you can identify that is you have this letter here and you have this letter up here. Now this one I tried to trick you. Instead of putting F's this time I put S's. So let's say you have an animal that shows this type of codominance, and its skin happens to be black and its skin happens to be white. Again, this letter can change and these letters can change. What happens if the skin was purple? That would also be acceptable. You just put the P up there. You put what you're talking about and then the actual color it is up there. Now that you'll get into certain things that aren't just dealing with skins or furs or feathers and colors, but for all our purposes, this is good enough for us right now. You can identify that this means that it is codominance. They can share. Now, can you tell me
which one of these do not mix, and instead you get one that shows a dominant and recessive. So you have a dominant and recessive Punnet chart, or Punnet square that you do. If you said number 1, then you're correct, because the dead giveaway is, first off, all these letters are the same. They're all D's, and then even this one's a lowercase d. That's a lowercase d, and so that should show you that there's a recessive and then dominant. Now, I could have tricked you and put an uppercase D right here too and asked you. Well, you might say, well, that's confusing because these are all uppercase now. But look at all the letters. All of them are the letter D. So that tells you that this is the recessive dominant. You're just looking at two people with all dominant traits. So the last one that it leaves you is the what. When these mix, what kind of dominance do we call that? If you said incomplete dominance, you are correct, because that red will mix with the blue to make purple. Now remember, when it mixes with purple, don't think that that RB comes together and changes its genotype to PP. No, it still keeps it as RB. Its genotype still stays like that, even though its phenotype is changed. Its genotype will still be that R and still be that B. Those letters will not change. Okay? So alright, if you have any other questions, make sure you email me, because the test is on Friday, and we'll go over all this stuff tomorrow in class, okay? So until next time, 6th graders, take care and good luck.